Finished up with two protozoan parasites that caused very different diseases in the human body. Trypanosome cruzi over here on the left-hand side, and Plasmodium falciparum, which is infecting red blood cells over here on the right-hand side. Both these organisms are able to evade the immune system, and that is that they can, once they infect a human, they can then go on to cause essentially what is would be a chronic disease, causing disease for many, many, many months or perhaps even years. The immune system is going to try to respond to it, but it's going to, in the end, be ineffective at clearing the infection from the human body. Both these organisms are able to infect human blood. However, the way they evade the immune system is very different. What Trypanosoma cruzi is doing is it's undergoing a process known as antigenic variation, where the microorganism essentially changes its coat, the way it appears to the human immune system after about a week or so being exposed to the immune system. And what this means is that after the initial infection has occurred and the immune system has responded to the presence of the organism then essentially becomes invisible, and the immune system needs to relearn what the pathogen is, generate a second immune system. And so what happens is there is a cycle of immune response followed by a cycle of antigenic variation, where the microorganism again increases in number over time. The little valleys that occur here are occurring because the immune system has gotten control of the organism initially. Once antigenic variation occurs, that immune response is no longer effective, and a new clone of the organism becomes very prevalent in the body. Plasmodium falciparum, on the other hand, undergoes a very different way of evading the immune system, simply by essentially hiding in plain sight. What happens with Plasmodium is it infects red blood cells, and we can see two red blood cells here which have been infected with Plasmodium protozoan. And when these protozoans are inside the red blood cell, they are essentially invisible to the immune system. And what happens is that patients become chronically infected with the immune system unable to reach the parasite. One last class of protozoans that we want to be able to know about, and these are the Amoebozoa. These can be very different in terms of their ultimate appearance. We see a very large freshwater protozoan called Amoeba proteus, but this is one that's going to be completely unable to infect a human. However, two that are of medical importance are over here on the right-hand side. Naegleria fowleri, which causes a extremely rare in the United States but extremely fatal disease called amoebic meningitis. This organism, Naegleria, is able to get into the central nervous system of an infected individual, causing a very severe form and generally fatal form of meningitis. One that is much more common throughout the world is infection due to this organism, Entamoeba histolytica, which causes amoebic dysentery. The microorganism is ingested through contaminated food or water, and then generally infects the intestinal system and then can migrate throughout the body. Both of these guys are going to be recognized by virtue of their amoeboid movement through tissue in a manner that's very similar to this large freshwater microorganism. These guys are going to end up being significantly smaller. So those are the protozoa. I talked about several with respect to case studies. Now we're going to move on to a new class of eukaryotic pathogens, and these are going to be members of the group of fungi. Fungi are eukaryotic organisms. They are going to have all the characteristics that other eukaryotes have, including all the typical organelles. Fungi are going to be adsorptive feeders. They're not photosynthetic, even though they are non-motile in nature. Fungi can be either a unicellular, in which case we refer to them as yeast, or multicellular, in which case we refer to them as molds. And unlike the protozoa, the fungi do all have cell walls. And the cell walls of fungi are going to be chemically very different from the cell walls of prokaryotes. The cell walls of fungi are composed of a chemical called glucan, which is a polymer of glucose, and a second polymer of carbohydrate called chitin. Chitin is a very common polymer that's of sugars that's found in many different organisms. The exoskeletons of arthropods, including insects, crustaceans, are all composed of this essentially indigestible carbohydrate polymer called chitin. Um, what makes fungi a little bit different from other eukaryotic organisms, in particular animals, is that their membranes contain a chemical called ergosterol, which helps to contribute to membrane fluidity. This is the same thing that cholesterol does in animal cells. We use the term mycoses to refer to fungal diseases, and although mycoses as a group can be a relatively significant health problem, there's fewer than 200 species of fungi which have been found to cause any sort of disease in humans. So essentially, most of the fungi are not going to be disease causing at all. We enjoy the benefits of yeast and other fungi and a variety of things throughout human society. We can see, of course, they make up a significant portion of our diet, and the ability of different microorganisms, including the yeast Saccharomyces cerevisiae, to produce important food-related biochemical modifications, such as the fermentation to produce alcohol, the process of fermentation to cause bread and other baked goods to rise. Other fermentations are important for lots of other food additives. The contributions of various yeasts and molds and mushrooms to human diet is extremely important. Other yeasts are incredibly important from the standpoint of biotechnology, and what we have demonstrated down here in the lower right is a simple experiment that was conducted by Alexander Fleming back in about the 1920s or so, where he observed that a colony of a fungus, a mold called penicillium, produced some sort of a chemical that apparently was able to keep microorganisms, other microorganisms, from growing. And that's what this zone of inhibition is surrounding this mold colony. What the mold colony turned out to be producing actually was penicillin, an antibiotic that is able to stop the growth of other microorganisms. And these antibiotics that have been identified from various yeasts and molds have been a huge importance for various forms of medicine. What's demonstrated down here in the bottom center is a microorganism called Neurospora. It's another mold that has an incredible role in the study of genetics. It's a very easy to study genetic system that allows the ready observation of mutants. It can be studied from a developmental standpoint, and a lot of the early work in genetics that has huge applicability to human disease was actually originally done in various species of fungi. And in fact, what's demonstrated over here on the left-hand side, this yeast genetic interaction network, we have actually for the model yeast Saccharomyces cerevisiae, the same guy that's responsible for beer and wine and bread. It has approximately 6,000 genes encoding many thousands of different proteins. And scientists have been able to figure out and describe the interaction of every single one of those genes with every single other genes. And it's just a fabulous tool for biotechnology, understanding on a very basic cellular level. We can then take those interactions that we can very clearly map out in yeast, a simple system, and use those to help us to understand what's going on in human cells as well. Here we can see a little bit better picture of what these unicellular yeasts look like and these multicellular molds. Some microorganisms, such as one of my favorite fungi, Candida albicans, is able to undergo a shift between a yeast form and a filamentous form, depending upon what type of growth conditions it's being grown under. Doesn't happen very easily with Candida albicans, but a number of the medically important fungi are the ones that are most significant from a human disease standpoint, actually very readily undergo this shift from a yeast form to a filamentous form and back and forth, depending upon the growth conditions. The major significant pathogens of humans are actually found in the yeast form when they're at 37 degrees centigrade, or in the temperature of the human body, and they are found essentially exclusively in the mold form when they're at temperatures cooler than the human body. Fungi are all adsorptive feeders, and what that means is that they like to secrete enzymes out into their environment, which help to break down nutritional sources that are out in the environment and then absorb those nutrients back into the body of the fungus. We can see that sort of illustrated down here. This mold we can see all around here is going to be secreting out enzymes which will digest nutritional sources out here in the environment, and then nutrients will be taken back up into the body of the fungus. Probably one of the most fascinating examples of this type of feeding we can see illustrated over here on the right hand side. This is actually a predatory fungus. That's a common soil microorganism, and it has little loops in its the body of the fungus and the body of these hyphae, and these loops can be used to trap small microscopic worms such as this nematode in these loops. Once this nematode becomes trapped by the body of the fungus then again secretes enzymes out into the environment. The enzymes kill and digest the nutritional sources within this nematode, and then these nutrients are then sucked back up into the body of the fungus as a source of nutrition. This mechanism of secreting out enzymes into the environment and then absorbing the nutrients is very characteristic of all the fungi. So let's look at a brief case study. This will be the last case study from chapter 12, which is describing an outbreak of coccidioidomycosis in travelers returning from Mexico. And this outbreak occurred in Pennsylvania in 2000. So the report describes an outbreak of coccidioidomycosis among travelers returning to Pennsylvania from a trip to Mexico on January 24th, 2035 church members from 2 cities in Pennsylvania traveled to Mexico, where they stayed for 1 week to construct a church. Within 2 weeks of returning home, 27 of the travelers complained of influenza-like symptoms. That would be fever and aches. Initial testing of acute serum samples at the Centers for Disease Control revealed antibodies to Coccidioidomys for one traveler. This is the causative agent of this disease, Coccidioides immitis. And how was this disease acquired? It was actually probably acquired from while they were doing the construction in Mexico. Coccidioides immitis is one of these dimorphic fungi that undergoes this switch from molds to yeast when it's at the warmer temperature. So at room temperature, it's going to be in the mold form. At 37 degrees centigrade, it's going to be in the yeast form. Well, it's not always in the mold form when it's in the environment. When it's in the ground, perhaps it may form spores, and the spores are going to be inhaled and are going to enter into the human body, where they then germinate into the yeast forms. Now, if we look at the distribution of Coccidioides immitis, it's not going to be a typical disease to find at all in Pennsylvania. And once this disease is diagnosed, what is going to help facilitate that diagnosis is actually looking at the patient history. This organism is endemic to the southwest United States and Mexico. And that's the big risk factor for these individuals in this case study for acquiring it. The risk factors are going to be traveling to areas where this fungus is going to be found in the soil and disturbing that soil and inhaling the spores. The clues to help diagnose the treatment are going to be the flu-like symptoms, which are indicative of a inhaled agent, as well as the location that all these patients were located at. Once the disease has been diagnosed, the recommended treatment is actually generally the same. Antifungals, such as amphotericin B, are going to be the recommended things to try to clear this up rather quickly. Now, the problem with many fungal infections is because they're eukaryotes, is that they are biochemically extremely similar to human cells. They're also many times typically kind of resistant to immune resolution and consequently can be responsible for causing kind of chronic infections that may be very difficult to treat unless they're treated appropriately with antibiotics. Because these cells are biochemically very similar to human cells, that kind of limits the types of antibiotics that we have. As a result, we have to use antibiotics that try to target some aspect of yeast biology that's different from human biology. One excellent target to try to go after is actually this membrane component, ergosterol. Ergosterol is something that's found in membranes that's going to be different from human membranes, and consequently, an antibiotic that targets ergosterol can do so without many serious side effects against human cells. Fungi reproduce either sexually or asexually, really kind of almost depending upon the mood that takes them and the availability of a fungus of the opposite mating type. Uh, as we kind of discussed in lecture already, yeast such as Saccharomyces cerevisiae are perfectly happy reproducing by the process of budding for essentially forever via a form of asexual reproduction. But if we bring two Saccharomyces cells that are of the opposite mating type, they can combine through a process of mating, generating a diploid cell that then is undergoing a process of sexual reproduction. Uh, it's actually during this process of sexual and asexual reproduction that many fungi become medically significant. Now, we can see in both these asexual cycles and sexual cycles of these molds that in one step, spores are released, and it's the spores that are actually pretty significant from a medical standpoint. They end up being rather aerobuoyant and can easily float along on air currents great distances, and this enables the spread of the organism during their sexual reproduction, but also allows them to be uptaken into the human body. All right, so that's the end of fungi. Let's talk a little bit about another group of eukaryotic cells, and those are the algae. The science of phycology is actually the study of algae. All the algae have something in common. They're all photosynthetic. They all have cell walls, and they all demonstrate many forms of sexual and asexual reproduction. But kind of like the protozoa, they're actually a rather large and diverse group of organisms. Not hugely significant from the standpoint of medicine with respect to the ability to cause disease. The only kinds of serious algae that are important with causing disease are ones that produce some different kinds of toxins that might get ingested accidentally and making somebody sick. But that doesn't happen hugely often. Algae are hugely important, though, for the cycling of nutrients throughout the biosphere as well as with food. But for those of us in microbiology class, our main interest in algae is actually the production of this chemical agar, which is produced from seaweeds. It's a carbohydrate polymer that is isolated from marine algae. It is, once it's been purified, is able to form a solid at low temperatures, at room temperature, at slightly elevated temperatures, but it dissolves and forms a liquid at high temperatures, such as the temperature of boiling water. Consequently, it can be used, it can be sterilized, and once it solidifies, once it cools, it can be used as a solid growth media for microorganisms to be able to grow on the surface of them. It's advantageous, though, in this use because there are extremely few microorganisms that are able to digest and break down agar. And so it makes a very nice inert substance for microorganisms to grow on. One last group of organisms that kind of ties together the algae and the fungi are the lichens. These are terrestrial algae. They're found on solid ground right here. We find them right here in York County if we look close enough. These are examples of organisms that are living together in a form of symbiosis, a symbiosis that we would actually describe with this term mutualism. In the body of a lichen, we actually have two different microorganisms living together. And algae, which are of course photosynthetic, grow in very close proximity to a fungal mass of hyphae. The algae are going to be photosynthetic and they're going to produce nutrients from the light of the sun that actually help to support the life of the fungus. The fungus is able to use some of the excess nutrients produced by the algal cells. The fungal mat, the fungal hyphae, helps to bring in water for the algal cells to grow and also offers a form of protection. We find here in very low magnification, visible, different forms of lichens growing essentially on solid rock. They're able to grow in very remote, very dry, very inhospitable environments. And it's only because these organisms are growing together in this form of mutualism that they're able to survive. The last group of important eukaryotes from microbiology class we want to talk to are animals. These, of course, are going to be multicellular organisms that lack, of course, a cell wall, and they're going to tend to be modal in essentially all cases. Most of the animals are actually not going to be microscopic at all. Animals that can actually cause human disease are organisms such as these helminths, these worms. This is a tapeworm, and you can see a little ruler down here for this guy by scale. You can imagine this guy growing actually inside the human intestine. We're not actually going to be able to see this organism except as parts of it break off and come through the back end of the digestive tract. Typically, the infections due to these helminthic organisms are going to be diagnosed by looking actually at fecal smears. Now, many of these organisms actually can live in a form of symbiosis with humans as well, in a form that we don't term mutualism, but we actually term parasitism. Fortunately, a good parasite is one that doesn't actually kill its host. And so, consequently, a human may actually have a very long lasting relationship with these. Once a helminthic infection is actually diagnosed, person's identified to actually have a tapeworm, antibiotics can be given that will eliminate this from the digestive tract, and health should go back to normal. Uh, these are going to be ingested via contaminated food, contaminated water, and they're going to be generally a nuisance more than anything else. We also speak about these organisms, insects and arachnids, which can bite human skin and can also be very important for transferring disease from one individual to another. And when we talk about insects and transporting of disease, we apply this term vectors. And many insects are hugely important for the transmission of disease throughout human populations. So hopefully this last set of videos here for chapter 12 has given a good summary of some of the eukaryotic classes of organisms that we care about from the standpoint of microbiology.